So movement and motility is almost synonymous with life. When you peer inside the inner life of a cell with a microscope, you can see that it's churning with moving objects. Many of these moving objects are small cargo containers called membrane organelles or vesicles. These are essentially the delivery trucks of the cell. At the time we were doing this experiment, 1984, we really didn't have answers to those fundamental questions of how that motility occurs. So our goal was to figure it out. So we figured if we were going to understand motility, we should look at it in a system which has the highest concentration of all of these motility components, these moving organelles, the motor proteins, and that's why we picked the axon. The axon is effectively a dedicated highway to molecular transport. A nerve cell, unlike a liver cell or many other cells, is extraordinarily long. One part of the cell may be in your spinal cord, but the other end of that same cell may be at the tip of your toe. And in between that cell body in your spinal cord and your toe is a very, very long tube that's called the axon that connects these very distal parts of the same cell. And this is essentially like shipping things between one part of the country and the other. Of all the axons that you can possibly choose, which one would you choose? Well, we decided to work on the squid giant axon. It is a much larger axon than an axon that you would find in a mammal, for example, or a human. We made the guess that this would be a good hunting ground to try to find which are the motor machineries that are causing this form of axonal transport. We decided to approach this problem by just taking apart the system in a test tube and see if we can put it together again and find out what were the critical components that were needed to get membrane organelles to move in this test tube environment. So what we did know were that microtubules were a key roadway for this transport of membrane organelles. So we knew how to prepare them from purified tubulin, and we can make very pure microtubule tracks. The next thing that we needed to get was the cargo that was moving inside the axon. These are membrane organelles. These we had to get from the squid giant axon. A third critical component was the fuel, adenosine triphosphate. That we knew was essential, and that we could fortunately buy from a chemical company. Most people, including us, thought that the motors would be tightly bound to the cargo. So the experiment was to make all these components, put them back together in this test tube environment, put it under a microscope, and see if we could see the movement of these membrane organelles occurring in this complete test tube in vitro condition, just as if it was occurring inside of the squid giant axon itself. This experiment was originally done in 1984 at an institute called the Marine Biological Laboratory at Woods Hole, Massachusetts. The day starts actually with fishermen going to sea and collecting squid and bringing them back to us. At that point, we have the animals in hand and we can begin our preparation. So for making the tubulin, we had already protocols that were developed for making tubulin from bovine brain, and we wanted to adapt it to the closest thing that a squid had to a brain, which is something called the optic lobe. So our strategy was to use the squid optic lobe as the biochemical source for squid tubulin, which we could then use to make microtubules. This part here is called the squid optic lobe. And I'm going to start by removing this squid optic lobe right there, which we'll use for our biochemical preparation of tubulin. From the squid, we wanted to then collect the giant axon. So this is the inside of the squid, and now we're going to do the dissection of the squid giant axon. And this is the cartilaginous backbone of the squid, which is called the pen. And you can see it looks like an old-fashioned writing pen. And I can also use this to point to different parts. So all of these lines that you see here are axons. They're all coming from this body of neurons right here. So all of these are innervating the muscle. And the biggest of these axons is the squid giant axon, and it lies right along the pen, right in the middle here. So this is the axon that we're going to have to dissect out, and there's one on either side here. So the next step is to dissect out that axon. We had to very carefully get the giant axon, tie it off, remove it from the squid. And here we are removing the giant axon, and also kind of a protective sheath that is covering it. There. And now we've got inside of here our giant axon. And we'll have to clean it up later, but that is the starting prep for removing it from the squid. So this is the squid giant axon right here. What I am doing is cleaning all of that sheath and kind of material that is covering the squid giant axon. Then we needed to get the inside of the squid axon out. That can be done fairly easily just by cutting it. And then you can squeeze out the inside of the giant axon more or less like rolling out toothpaste from a tube. And that rolled out axoplasm, which is the inside of the squid axon, was the starting point for our preparation. From there, we had to then prepare the cargos, the membrane organelles, and separate them from the soluble protein fraction. So inside a cell, many proteins are bound to particular compartments, a mitochondria, a nucleus, a membrane organelle. But there are also many proteins that are freely moving around in the cytoplasm. They're unattached to anything. And these are often called cytoplasmic proteins. And when you break open the cell, they end up being in what's called a soluble protein fraction. So to do this, we took the axoplasm, put it in a buffer, which is a biochemical solution, disrupt the axon with a pipetteman, and then we place that disrupted axoplasm on something called a sucrose gradient. A sucrose gradient is a biochemical technique that can be used to separate, or another word is fractionate, different components inside of a cell. Imagine taking sucrose, sugar, which may be in your kitchen, and dissolving it in water. But now keep adding more and more sucrose. Eventually, it gets very viscous and thick. That was the general solution that we used in this sucrose gradient, a very thick, viscous solution of sucrose. In fact, we made different percentages of sucrose. A very dense layer at the bottom and progressively lower density layers higher up in this tube. We then put the axoplasmic fraction on top of this sucrose gradient and then put it in a centrifuge which would spin that tube around amazingly fast. So here I'm going to put our sample in the bucket here. And it's very important that you have a counterweight. In fact, these two buckets have to be very carefully weighed so the rotor doesn't become imbalanced. I'm going to put it now inside the centrifuge. What happens then is all the material in that sample start moving down the tube through the sucrose gradient. If you're a very large object, you travel down that tube faster. And that causes the membrane organelles to collect there and form a little band between that interface between the low and the high density sucrose. And this allows us to get a very crisp band of those membrane organelles that we could remove with a pipette and then use in our experiments. But one thing you learn in biochemistry is to keep all of your fractions when you do an experiment, whether you think you may need them or not. In fact, we kept other parts of that sucrose gradient, including one fraction which is at the very top of the sucrose gradient, which was the soluble protein fractions. And again, we just put that aside and also placed it in the ice bucket, just in case we needed it later. Now we had all the ingredients. We had purified microtubules made from purified tubulin. We had the membrane organelles. We had the chemical energy source, ATP. And now we can add them in various combinations and see what might work to allow this magical process of organelle transport to occur in the test tube. We found that when we added organelles, microtubules, and ATP, surprisingly, there was no movement. But we found that the organelles themselves were not capable of moving until we added this soluble protein fraction. And finally, we saw tremendous movement of these organelles along the microtubules. This was a really exciting moment. I'd been working essentially for 4 months to get these organelles to undergo motility in a test tube just like in the native Axon. But the next experiment was even more surprising. It was called a control experiment. Essentially, it wanted to make sure that what we thought we saw moving were in fact the membrane organelles and not something else that might have been coming along with the soluble protein fraction. So the way to figure that out was just to mix everything again, but leave out the membrane organelles. Just add together the microtubules, the ATP, and the soluble protein fraction. And we thought nothing would happen, that we wouldn't see anything, and then we can call it a night and go home. But what happened was really mind-blowing. We looked under the microscope, and the microtubules, which are these long biological polymers, were sliding around, crawling around like snakes on the glass surface. That was something that we certainly didn't expect at all. And in fact, it even took us a few minutes to even think about what was happening here. Why were these microtubules crawling along the glass surface? After we overcome our amazement and surprise by that result, we then began to piece together in our head what might be happening. Maybe that soluble protein fraction that contained some agent that was causing the organelles to move actually contained the motor itself. So the soluble protein fraction had motors that were floating around in it without the organelles, and these motors would also be able to stick onto the surface of this glass slide, grab hold of the microtubules, and transport these microtubules along the glass. Think of a rock concert. The singer jumps off the stage into the mosh pit, and the people in the audience who are stationary there grab hold of the singer and transport the rock star across. We thought a very similar thing was happening there with the microtubules crawling on glass. The next day, I was able to extend those results to find that we could get plastic beads in the presence of this soluble axoplasmic protein fraction also to move along microtubules. And this is sometimes what happens in science. You know, you work really hard, you don't get any results, it gets frustrating, you have to persevere, and then one magical day, everything really seems to come together, and the experiment is suddenly propelled much farther than you could have ever imagined. And that's a little bit the nature of science. It's when persistence counts for a lot. One has to be prepared for the unexpected. And one has to believe that one's experiment is going to work at the very end. And I think once one has that kind of experience, one's also willing to persevere on a lot of other experiments later in one's scientific career as well. I stayed on at Woods Hole over the winter to purify the first motor of the Kinesin superfamily, which was a new motor protein at the time. We now know that kinesin is not one motor, but it's a large family of related motor proteins. For example, in humans, there are 45 different genes encoding different kinesin motor proteins. And there's so many because they're involved in many different kinds of motor activities, not just axonal transport, which first motivated this study, but a variety of other cargo transport and even activities in cell division, such as the construction of the mitotic spindle and the separation of chromosomes. These motility experiments really are what opened up the field. It first of all opened up the field by allowing us to be able to detect this activity of a motor protein that was critical for the subsequent purification. In many ways, the purification is technical. We had biochemical techniques that were already well established that we could use to go about and purify a protein. So these in vitro motility experiments really provided the foundation of being able to assay and detect the activity of a motor protein that we could then apply to the purification of a kinesin. But it was much more than that. These in vitro motility experiments, kind of gave us this window into this life of what a motor protein is actually doing and how it might be working. It kind of gave us an experimental tool that we just didn't have before to study motility. I mean, before we could just look at cells with a microscope a little bit helplessly, seeing all this motility going on, but not knowing really how to dissect it or how to understand it or how to experimentally manipulate it. It was fascinating, of course, but we really couldn't get at the basic questions of how these machines might actually work. These in vitro motility experiments essentially opened up a whole new frontier of how we can measure motor proteins.